Hello, in this topic, we will be learning about the cell, the unit of life, and in that, we will discuss about the prokaryotic cell. So, first of all, for learning the prokaryotic cell, we should know what is cell and what are the types of cells. So, cell is the basic structural and functional unit of life. Okay, so why it is called basic structural and functional unit of life? For example, a wall. Wall is made up of what? It is made up of number of bricks. Number of bricks they join to form the wall. Okay. So, and different wall forms the room. Different room forms the home, house. And different house form the floor. And different floors form the building. Okay. So building, imagine building is the organism. Room is the house is the organ systems, and floor is the organ system. Many houses in the floor, that is house organ system. Then the room is the organs, wall is the tissues, and the bricks are the cells. So in this manner, we can learn what is cell. So similarly, cell is also made up of uh, made up of atoms. Atoms join to form a molecules, and this molecules, many molecules form the cell. Cell forms the tissue. Tissue forms the organ, and organ form the organ system. Many organ system forms the organism. So that's why it is called as the basic structural and functional unit of organism. Now, based on the number of cells, the organism are classified into unicellular and multicellular. Unicellular organism are the organism which have only one cell, which is made up of only one cell. Whereas multicellular organisms are made up of many cells. Made of many cells and made of unicellular, made of single cells. So now how we came to know that the cell is existence, how we came to know the existence of cell. It was the scientist Robert Hooke who discovered the cell from a sample of a cork. Cork is nothing but a bark of a tree. He observed a honeycomb-like structure in the microscope. This honeycomb-like structure appears like a small compartments. There were many small compartments like structure was present. So each compartment was appearing like a little room, which he called it as a cell, because in Latin the cell word came from the Latin word cellula, which means cell. Okay, so it is appearing like a little room, so that's why it gave the term cell. So since then till now, the cell word is used for the basic structural and functional unit of life. We call it as a cell. Then there was a scientist, Anton von Leeuwenhoek. He observed firstly the living cell, first time the living cell he observed. Then there was a scientist, Robert Hooke, who discovered the nucleus are present in the cell. Then there was a two scientists, Sleden and Schwann. They both studied the plants and animal respectively. So what they came into conclusion that plants and animals are made up of many cells, and which group to form the tissues, and ultimately they form the organism. So they came up with the hypothesis that organisms are made up of cells and product of cells. But there was some drawback in the theory. they did not explain how the new cells are formed. It was Rudolf Virchow who gave the cell theory, who completed the cell theory that the new cells are formed by dividing the pre-existing cell. The cell divides to produce a new cell. So there is a division of cell, which leads to the formation of new cells. So it arises from the pre-existing cells. Now we have learned the cell about cell in previous class 9th and 10th standard. So there is a onion peel we have learned and human chick cells. So there are three basic structures observed in cell, such as cell wall, cell membrane, okay, cell membrane, nucleus, cytoplasm. These three structures are observed almost in every cell. Cell membrane, cytoplasm, and the nucleus. The human chick cell lacks the cell wall, whereas onion peel cell have the cell wall because it is a plant cell. Then with the advancement of microscope, we came to know that there are other organelles present in the cell, such as nucleus, Golgi complex, microtubules, endoplasmic reticulum, centrioles, lysosomes, mitochondria, ribosomes, etc. that we came to know with the advancement of microscope. So there are two types of cells, prokaryotic cells and eukaryotic cells. The organism which have prokaryotic cells are called prokaryotic organism, and the organism which have eukaryotic cells are called eukaryotic organism. So prokaryotic cells they don't have a well-developed nucleus. They don't have a nuclear membrane. Nuclear membrane is absent. Only the nucleoid structures are present. This network-like coiled structure of nucleus is present inside the cytoplasm, embedded in the cytoplasm. So that, and there are also they lacks the membrane-bound cell organelles. Okay, whereas in eukaryotic cell they have a well-developed nucleus and a membrane-bound cell organelles. Cells are of different size and shape. Microplasma, it is the smallest cell. It is 0.3 micrometer. Then bacteria is 3 to 5 micrometer. Human red blood cells is 7 micrometer. And nerve cells are longest, up to 1 meter to 2 meter length. It is there in the body of animals and humans. And largest cell is the egg of ostrich. So these are the different size. Okay, the cell varies in size in different organism. Then shape, it also varies in shape. There is a columnar shape, there is a cuboidal shape, there is a elliptical shape, elliptical biconcave shape. Okay, so it is a RBC cell, and there is a spindle shape. There is a network-like shape, and there is a irregular, you can say irregular shape cells. So the shape of the cell is varies according to their function. So that we will discuss in the tissue chapter. And now we will learn about the prokaryotic cells. So bacteria, prokaryotic cell consists of the bacteria, blue-green algae, mycoplasma, and PPLO, that is pleuropneumonia like organism. So these bacteria and these prokaryotic cells are very tiny and small. They are unicellular and they multiply very rapidly than the eukaryotic cells. They vary greatly in shape and size. So there are different shapes. There are four basic shapes observed in bacteria such as coccus, bacillus, vibrios, spirelia. So this four basic type of shape and size of bacteria are observed in prokaryotic cells in bacteria. Now structure of a typical bacterial cell. This bacterial cell is having a wall. cell wall we can say, or the membrane, okay, outer wall. So this outer wall is composed of three layers. The first layer is the capsule, second layer is the cell wall, and third layer is the plasma membrane. Each wall have a distinct function. Capsule performs the function of protection. Cell wall function as giving a shape to the cell. Plasma membrane function as a selectively permeable. Okay. Inside the plasma membrane in the inner side of the bacterial cell, they form an extension. They form extended structures called as the mesosomes. This mesosomes perform a very essential function of the bacterial cell. These mesosomes are responsible for the formation of different molecules for the cell wall formation. Then it is responsible for DNA replication, then respiration, and several enzymatic reaction of the cell. Also, there are in cyanobacteria, this plasma membrane structure this mesosome structure have a chromatophores which contain the pigments. We say now bacteria have the bacteriochlorophyll. Bacteria do not have the chloroplast. As we have learned that prokaryotic organisms do not have the membrane-bound organelles. So they don't have chloroplast. So the bacteriochlorophyll is present in the membrane, inner membrane of the, you can say, bacterial cell. Then there is a fluid present, that is the cytoplasm. And there is a coiled structures, that is a chromosomal structures, that is called as the nucleoid. This nucleoid is made up of DNA. So there is no membrane. It is not present inside the membrane. It is present naked in the cytoplasm. So nucleoid, that structure is called nucleoid. Then there is a extra chromosomal DNA. Apart from nucleoid, there is another extracircular DNA in the bacterial cell, that is plasmid. This plasmid is having another important role in the bacterial cell. It have certain features which protects the bacteria, such as antibiotic resistant site. Okay, so it helps the bacteria to resist from various antibiotics. Okay, so this is one example. So this plasmid is useful for, in biotechnology we will learn that this plasmid is useful for recombinant DNA technology, in which we can introduce the foreign DNA and we can recombinate the DNA. Then there are inclusion bodies. Inclusion bodies are the reserve materials of the cells, such as phosphate granules, then glycogen granules, cyanophycin granules. So these are the different reserve materials of the cells are present in the form of granules, which are called as inclusion bodies. Then there is a ribosomes. Ribosomes is present in both prokaryotic and eukaryotic. It is the only organelle which is present in both prokaryotic and eukaryotic organs. And it is not membrane bound. In bacteria, there is a 50S and 30S type of ribosome. 30S is the smaller subunit, 50S is the larger subunit. They together form the 70S type of ribosome in bacteria. Okay. Then there is a pilus. Single is called pilus and many is called pili. Okay. So this pili helps the bacteria. It does not help the bacteria in locomotion. It helps the bacteria in attaching with the surface or attaching with the host tissues. So it is for that purpose. Then it have, if the bacteria is motile, it have the flagella. Okay, which helps in the locomotion. So that's all about the prokaryotic cell, that is bacterial cell. Thank you.